Welcome friends to a new session on molecular biology. In this session, we will discuss the initiation, elongation and termination of transcription in prokaryotes. According to central dogma of molecular biology, genetic information flows from DNA to DNA during its transmission from generation to generation and from DNA to protein during its phenotypic expression in an organism. The transfer of genetic information from DNA to protein involves two steps. 1. Transcription. It is the transfer of genetic information from DNA to RNA. And 2. Translation. It is the transfer of genetic information from RNA to protein. During transcription, one strand of DNA of a gene is used as a template to synthesize a complementary strand of RNA called gene transcript. The basic features of transcription are the same in both prokaryotes and eukaryotes, but some features like promoters are different. The RNA polymerase of E. coli are studied in great detail. This enzyme catalyzes all the reactions in RNA synthesis. There are about 7,000 copies of RNA polymerase in an E. coli cell, and majority of them are involved in RNA synthesis. A DNA segment that is transcribed to produce one molecule of RNA is called a transcription unit. Transcription of genes may include several contiguous genes, or they may be equivalent to individual genes. In bacteria, large transcripts that carry the coding sequences of several genes. The process of transcription in prokaryotes can be divided into three stages. 1. Initiation of new RNA chain. 2. Elongation of the RNA chain. 3. Termination of transcription and release of the nascent RNA molecule. The transcription process in prokaryotes is catalyzed by a complex enzyme, RNA polymerase. The E. coli RNA polymerase has a molecular weight of about 4,80,000 Daltons and consists of 5 polypeptides, including 2 chains of alpha polypeptide, 1 chain each of beta, beta prime and sigma polypeptides. The RNA polymerase molecule, the holoenzyme, has the composition alpha 2 beta, beta prime, sigma, in which attachment of sigma factor is not very resistant, so that the core enzyme alpha 2 beta, beta prime can be easily isolated. The alpha subunits are involved in the assembly of the tetrameric core of RNA polymerase. The beta subunits contains the ribonucleoside phosphate binding site, and the beta prime subunit harbors the DNA template binding region. One subunit, the sigma factor, is involved only in the initiation of transcription. It plays no role in elongation. After RNA chain initiation has occurred, the sigma factor is released and chain elongation is catalyzed by the core enzyme. The function of sigma is to recognize and bind RNA polymerase to the transcription initiation or promoter sites in DNA. The core enzyme, where sigma is absent, will catalyze RNA synthesis from DNA templates in vitro. In so doing, it will initiate RNA chains at random sites on both strands of DNA. In contrast, the holoenzyme, where sigma is present, initiates RNA chains in vitro only at sites used in vivo. Promoter sites for initiation of transcription in prokaryotes. For synthesis of RNA on DNA template, there are transcription units, each having a starting point and a termination signal. It is not necessary that the RNA may be released at the termination signal, since there are situations where transcription may continue beyond the termination signal due to anti-terminator molecule, as in the case of lambda phage. Biologists often use upstream and downstream to refer regions located toward the 5' end or 3' end respectively. Upstream or downstream to the start point of a transcription unit, there is always a DNA sequence which is necessary for the transcription to start. This is called a promoter site, which is cis dominant or cis acting, since it is not involved in the synthesis of any diffusible substance. It means that the promoter site should be physically continuous with the transcription unit to be transcribed and cannot function if present on another chromosome within the same cell. The binding sites for RNA polymerase enzyme lie within the promoter sequence and may be 41 to 44 base pairs in length in E. coli, where more than 100 promoters have already been sequenced. There is hardly any long conserved sequence over majority of the promoter regions, although short sequences within the promoters are conserved. The start point is a purine in over 90% cases, and upstream from this start point is a 6 base pair region TAT, which is called Pribnov box and is available in almost all promoters. The conservation of these 6 bases varies from 45 to 96% as represented by the subscripts in the following sequence: T80, A95, T45, A60, A50, T96. The center of Pribnov box lies usually 10 base pair upstream, and therefore it is sometimes described as -10 sequence, although the actual center varies from -18 to -12. There is another sequence with the following: T82, T84, C78, A65, C54, A45, which is lying -35 bases upstream and is called recognition region. Although details of binding reaction are not known, typical promoters in E. coli use -35 and -10 sequences to be recognized by RNA polymerase. It is obvious thus that a typical promoter in bacteria has 3 components: 1. Consensus sequence at -10 base pair, 2. Consensus sequences -35 base pair, and 3. Start point. Initiation of new RNA chain. The RNA synthesis or initiation of new RNA chain by RNA polymerase enzyme proceeds in 3 steps. First, the holo enzyme binds at the promoter region in DNA, forming the closed promoter complex in which DNA remains double helical. Secondly, the closed promoter complex isomerizes and causes unwinding and separation of DNA strands to form open, that is binary promoter complex by RNA polymerase enzyme, providing a template strand free to base pair with incoming nucleotides. Finally, the formation of a phosphodiester bonds between the first few ribonucleotides in the nascent RNA chain takes place. After unwinding the DNA double helix, only one of the 2 strands is copied. This is achieved by incorporation of nucleotides initially without movement of RNA polymerase enzyme, leading to the formation of RNA chain up to 9 bases in length. During the incorporation of these 9 bases, at every step there is a possibility for the release of this small RNA chain by a process called abortive initiation. It describes a process in which RNA polymerase starts transcription but terminates before it has left the promoter. It then reinitiates. When RNA polymerase binds to a promoter, it separates the DNA strands to form a transcription bubble and incorporates up to 9 nucleotides into RNA. There may be a cycle of abortive initiations before the enzyme moves to the next phase, elongation. After the first bond is synthesized between 2 ribonucleotides, the RNA polymerase must clear the promoter. During this time, there is a tendency to release the RNA transcript and produce truncated transcripts. This is called abortive initiation and is common for both eukaryotes and prokaryotes. Once the transcript reaches approximately 23 nucleotides, it no longer slips and elongation can occur. This is an ATP dependent process. A cycle of abortive initiations usually occurs, generating a series of short oligonucleotides consisting of 2 to 9 base before initiation is actually successful. As mentioned earlier, the sigma factor of RNA polymerase mediates its binding to promoters in DNA. In E. coli, hundreds of promoters are sequenced and found to have little in common. In E. coli, 2 short sequences within these promoters are sufficiently conserved to be recognized. The midpoints of the 2 conserved sequences occur at about 10 and 35 nucleotide pairs respectively before the transcription initiation site. Thus, they are called -10 and -35 sequence respectively. The nucleotide sequences that are present in the conserved genetic elements most often are called consensus sequences. The -10 consensus sequence in the non-template strand is TAT-AAT. The 35 consensus sequence is TTGACA. The sigma factor initially recognizes and binds to the -35 sequence. Thus, this sequence sometimes called recognition sequence. The AT-rich -10 sequence facilitates the localized unwinding of DNA double helix, which is an essential prerequisite to the synthesis of a new RNA chain. In E. coli promoters, the distance between the -10 and -35 sequences is highly conserved, never being less than 15 or more than 20 nucleotide pairs in length. Once initiation succeeds, the sigma factor of RNA polymerase dissociates. The dissociation of sigma factor marks the entry of NAS-A protein, which helps the elongation and promotes pausing and termination of transcription at specific sites. The core enzyme now undergoes a major conformational rearrangement and a stable ternary elongation complex is formed. The elongation complex moves along DNA, synthesizing RNA all along its path at a rate of about 40 bases per second at 37 degrees centigrade. RNA polymerases in some phages like T3, T7 are each a polypeptide which is less than 1 lakh Daltons and which synthesizes RNA very rapidly, that is approximately 200 bases per second at 37 degrees centigrade. Elongation of RNA transcript continues till an unstable termination complex is formed. Elongation of the RNA chain. Elongation of RNA chain is catalyzed by the RNA polymerase core enzyme after the release of the sigma subunit. The extension of RNA chains takes place within the transcription bubble locally unwound segment of DNA. The RNA polymerase enzyme contains both DNA unwinding and DNA rewinding activities. RNA polymerase enzyme continuously unwinds the DNA double helix ahead of the polymerization site and rewinds the complementary strands behind the polymerization site as it moves along the double helix. The average length of transcription bubble in E. coli is 18 nucleotide pairs, and about 40 ribonucleotides are incorporated into the growing RNA chain per second. As RNA polymerase moves along the DNA molecule, the nascent RNA chain is displaced from the DNA template strand. Inchworm model for elongation of transcript. In the year 1993, M.J. Chamberlain proposed that the transcript elongation involves inchworm-like translocation of RNA polymerase along the DNA template. RNA polymerase is assumed to have one site for DNA binding and two sites for RNA binding, the leading product site and the lagging product site. While the leading product site gets filled about 10 newly added nucleotides in each successive cycle of elongation, the lagging product site occupies the preceding segment of nascent RNA earlier synthesized. RNAP translocates forward by about 10 nucleotides when the leading site is full, so that the leading site is emptied again, thus permitting the next cycle of elongation. Stability to the elongation complex is provided by the interaction of RNA with the lagging site of RNAP. The Inchworm model is based on experimental evidences. Termination of transcription and release of the nascent RNA molecule. The elongation of the transcript may be impeded by 1. Pause sites, 2. Arrest sites or dead ends, and 3. Terminators or release sites. The pause sites induce a temporary reversible block to nucleotide addition. The arrest sites stop elongation to be resumed only in the presence of elongation factors GRE A and GRE B. In eukaryotes, the corresponding transcription factors is TF2S, which assist in a novel transcript cleavage reaction. The release sites leads to release of RNA and RNAP either intrinsically or upon activation by factors like NAS-A, Rho or TAU. Although the nature of DNA sequences causing transcription arrest is not known, the mechanism of transcription arrest and the roles of GRE-A and GRE-B in restoring elongation can be explained in the framework of Inchworm model described earlier. The transcription arrest may result when the catalytic site of RNAP is misplaced from the 3' end of growing transcript. This itself may result if the lagging product site slipped backward on the template, bringing the catalytic site out of register. Transcript cleavage by GRE-B would create a new 3' end into the right register, thus restoring elongation. Further, at some template sites, translocation may disturb the placement of catalytic site into the right register, and GRE-A induced cleavage of short segments might restore the appropriate register to resume elongation. Thus, an elongation barrier may be overcome by RNAP through a backup and restart mechanism. Termination of RNA chains in prokaryotes occurs when RNA polymerase encounters a termination signal on DNA. When this occurs, the transcription complex dissociates, releasing the nascent RNA molecule. These DNA sequences providing termination signal are called terminators. However, sometimes termination event despite the presence of a terminator can be prevented by some factor interacting with RNA polymerase. This phenomenon is called anti-termination, and the factor responsible for this phenomenon will be called anti-terminator. It will be seen that the termination actually depends on the scrutiny of not only the DNA template, but also that of the product, that is RNA, obtained by transcription. There are two types of transcription terminations in E. coli. The termination signals found on DNA can be of two types. One, those which are recognized by a protein factor known as Rho factor, which cause Rho dependent termination, and two, those which do not need Rho factor for termination, which cause Rho independent termination. Rho independent termination of E. coli. Rho factor is a protein of molecular weight 55,000 Daltons and was isolated from E. coli. It causes termination of most mRNA molecules in vitro at relatively low ionic strength, but is not needed for termination of some of the mRNAs. Rho independent terminators contain a GC rich region followed by 6 or more AT base pairs. The nucleotide sequence of the GC rich region is such that the regions of the single stranded RNA can base pair and form hairpin like structures. This hairpin like structures forms immediately after the synthesis of the participating regions of the RNA chain and retard the movement of RNA polymerase enzyme along the DNA, causing RNA polymerase to slow down or cause a pause in transcription. Since adenine uracil base pairing is weak, requiring less energy to separate the bases than any of the other stranded base pairs, the run of uracils after the hairpin region is thought to facilitate the release of newly synthesized RNA strand from the DNA template when the hairpin structure causes RNA polymerase enzyme to pause at the site. At Rho dependent terminator, Rho protein is active as a tetramer. A model for its action proposes that Rho binds to the 5' end of nascent mRNA chain and moves along the length of mRNA. When RNA polymerase is having a pause due to hairpin structure, Rho catches up, interacts with RNA polymerase and releases the mRNA molecule from template, accompanied with the dissociation of RNA polymerase and Rho factor. Rho dependent termination of E. coli. The mechanism by which Rho dependent termination of transcription occurs is still uncertain. Rho dependent sequences are 50 to 90 base pairs long, rich in cytosine residues and largely devoid of guanines. In Rho dependent termination, the Rho protein binds to the growing RNA chain and moves from 5' to 3' along the RNA, seeming to pursue the RNA polymerase enzyme, catalyzing the synthesis of RNA chain. When RNA polymerase slows down or pauses at the Rho dependent termination sequence, Rho catches up with the polymerase enzyme and pulls the nascent RNA chain from the transcription bubble. Evidence for termination by the Rho protein is that Rho protein hydrolyzes ATP in the presence of single stranded RNA, but not in the presence of DNA or duplex RNA. Hexameric Rho, which is structurally similar to and homologous to ATP synthase, specifically bind single stranded RNA. A stretch of 72 nucleotides is bound in such a way that the RNA passes through the center of the structure. Rho protein is brought into action by sequences located in the nascent RNA that are rich in cytosine and are poor in guanine. The ATPase activity of Rho enables the protein to pull the nascent RNA chain while pursuing RNA polymerase. When Rho catches the RNA polymerase at the transcription bubble, it breaks RNA DNA hybrid helix by functioning as an RNA DNA helicase. Additional information about the action of Rho protein was obtained by adding this termination factor to an incubation mixture at various times after the initiation of RNA synthesis. RNAs with the sedimentation coefficients of 10s, 13s and 17s were obtained when Rho was added at initiation, a few seconds after initiation and 2 minutes after initiation respectively. If no Rho was added, transcription yields 23S RNA product. It is evident that the termination contains at least 3 termination sites that responds to Rho yielding 10s, 13s and 17s, and 1 termination site that does not respond to Rho yielding 23S RNA. Rho independent terminator differs from Rho dependent terminator in the presence of a long run of uracil residues in mRNA and adenine residues in DNA, and of a GC rich region in stem of hairpin structure formed by mRNA and AT rich structure in DNA. Let us summarize the following discussion on transcription, initiation, elongation and termination in prokaryotes. A DNA segment that is transcribed to produce one molecule of RNA is called a transcription unit. RNA synthesis occurs in 3 stages: initiation of RNA chain, elongation of RNA chain and termination of transcription and release of nascent RNA molecule. The RNA polymerase enzyme that catalyzes the transcription is complex multimeric proteins containing 5 polypeptides. The elongation of RNA chain occurs within the transcription bubbles, locally unwound segments of DNA. Elongation of RNA chain stops when RNA polymerase enzyme encounters transcription termination signals in DNA. There are 2 types of transcription terminators: Rho dependent termination and Rho independent termination. Rho protein is an ATP dependent helicase that binds the nascent RNA chain and pulls it away from RNA polymerase and the DNA template. Now, I will give you certain assignments based on the discussions what we had. 1. Briefly explain the overall mechanism of protein synthesis. 2. Write short notes on transcription unit. 3. Explain the process of transcription. 4. Discuss Inchworm model of elongation. 5. Give a brief note on Rho dependent and Rho independent termination. I will give you certain references for your further reading. The first one is a book with the title Biochemistry 5th edition, authored by Berg, Timsko, Stryer in the year 2008. It is published by Freeman and Company, New York, USA. A second book is with the title Cell Biology 7th edition, authored by Karp, published by John Wiley and Sons, Singapore. A third book is with the title Molecular Biology of the Cell 5th edition, authored by Alberts, Johnson, Lewis, Raff, Roberts and Walter in the year 2007, published by Garland Science, New York and London. Next one is a book with the title Gene 9, authored by Levin, published by Johns and Bartlett Publishers, Sudbury, Massachusetts. Yet another book is with the title Biochemistry, authored by Davids and Sitman in the year 1997, published by Waverly, New Delhi. Yet another book is with the title Principles of Genetics, authored by Gardner and Nustad in the year 1984, published by John Wiley, New York. Another book is with the title Principles of Genetics, authored by Sinnett, Dunn and Dobzhansky in the year 1958 and published by McGraw-Hill, New York. I will also give you certain websites for a further reading. First one, www.bioencyclopedia.org. Second one is www.oxfordgenetics.com. We have come to the end of this session. We will meet in another session with a new topic. Until then, bye.